Hello, my name is Immo Scheffler. As Professor of Biology at UCSD and as the holder of the Paul D. Saltman Chair in Science Education, I would like to welcome you to Science Matters. It is my distinct pleasure to dedicate this series to the memory of Professor Paul Saltman. Paul's passion for science was exceeded only by his passion to communicate about science. We hope to follow in his footsteps in bringing you these presentations so that scientists at UCSD can share with you the excitement of discovery and emphasize the relevance of modern biology to society and especially to you, the citizens of our community. I hope you will enjoy this presentation of Science Matters.
I bring to you today a simple idea that has a lot of very interesting ramifications for the future. And the idea is that modern biology and genetics is based on the understanding of a molecular language contained within cells. From an alphabet of only 4 letters comes a mute but eloquent literature whose highest expression is human existence. What I'd like to do then is review the story of some key discoveries over the past 100 years that have led us to where we are today, talk a bit about the nature of this molecular language, and then reflect on what it means for 21st century biology and healthcare. I'm honored to give this talk in the biology department, and as you see, come from the School of Medicine. As a result, most of the examples I'm going to use relate to human health and disease. It can't be helped. It's just the way doctors think about things. They're always just constantly thinking about their own species. But you should understand there's a whole world of interesting biology out there that involves plants and animals and tiny little microbes. And the important message is that at the molecular level, living things are much more alike than they are different. And things we have learned about bacteria and yeast and worms and mice continue to teach us quite a bit about this human organism, and vice versa. So in the spirit of this cartoon, we're going to begin on the ground floor and hopefully help you to get to the next level for future talks that you'll hear in the Science Matters series. Now, the traditional science of genetics, out of which modern molecular biology and molecular genetics has grown, is really itself quite young. Plant and animal breeders had practiced their art for centuries, but it was only in the last half of the 19th century that the monk Gregor Mendel conducted his very careful crossbreeding experiments with smooth and wrinkled peas and other plants. The laws of inheritance that he proposed based on his observations went largely unnoticed until about 1900, when they were rediscovered. The English biologist William Bateson is usually credited with inventing the term genetics that appeared for the first time in the Oxford English Dictionary in 1905 to describe the science of understanding inheritance. Now, throughout the first half of the 20th century, the description of the behavior of inheritance in living cells was described in somewhat exempted and mystical terms. That is, the process of living was not subjected to this reductionist approach of taking complicated systems apart to see how the parts worked. The notion that predictable and ordinary chemical processes lay at the foundation of living cells was simply not a part of most biologists' mindset. Laws that had been developed for physics and inorganic chemistry were believed to be irrelevant to the privileged existence of living things. The chemical makeup of cells was, however, known. The most likely candidates to account for heredity were proteins. They were the largest and most complicated molecules known, and most scientists believe that only proteins could contain enough information to make other proteins. The light of a dawning age appeared in 1943 with a publication by Oswald Avery and his colleagues that the inheritance of biological behavior or biological properties could be transmitted by a substance called deoxyribonucleic acid, or DNA. He showed that one could take the DNA from infectious bacteria that had been killed and insert it into harmless bacteria, and the harmless bacteria would then be able to cause infections in mice. The significance of Avery's work was widely disputed in the 40s, and only later was it confirmed from other experimental systems that people sort of grudgingly admitted that DNA might be the molecule of inheritance. Still, how it transmitted biological function was really a puzzle, since it seemed to have very little to distinguish itself. It remained for the English biologist Francis Crick and his American postgraduate student James Watson to assemble the known biological and crystal data about DNA into the now familiar double helix model that was published in Nature in 1953. Theirs was the first model that specified that the information content of DNA was encoded in the sequence of a spiral chemical structure, not primarily in the relative ratios of its component parts. Now, the information encoding part of DNA is represented by an alphabet of only 4 letters. Chemical subunits called respectively adenine, guanine, cytosine, and thymine. These nucleotide bases, as they are called, are stacked like coins within this spiral staircase, a backbone made up of deoxyribose, a 5-carbon sugar. Now, this structure provided for the first time a ready explanation of how cells might reproduce themselves and their genetic information. The double helix uncoils, and a new strand of so-called complementary bases lines up based on attractions and repulsions between similar molecules. Now, shown in this little movie, the notion that if one opens the double helix, that nucleotide base pairs will automatically and with very high precision find their complementary base. So A's always pairing with G's and CT's, and adenines always pairing with thymines, and guanines always pairing with cytosines. This process, in fact, has an error rate of, in living cells, of only about 1 in a billion times does that process go bad. But DNA doesn't exist just to reproduce itself. The major function of this thread of life is the control of the synthesis of proteins. Some proteins are the structural building blocks of the cells that make up organisms, and some of them function as enzymes, controlling chemical processes of energy production and distribution in the cell. In order to make proteins, DNA is copied into an intermediate molecule called ribonucleic acid, or messenger RNA, that is very similar in structure, and this process is known as transcription. The information is transcribed into this other nucleotide molecule. That molecule migrates out of the nucleus of the cell and then is translated into a corresponding sequence in a final protein that is produced, the protein being made up like beads on a necklace of units called amino acids. This is the process called translation. That is, the information content of the nucleotides is translated into another environment of protein sequence. The DNA and messenger RNA code is read in groups of 3 bases each. This is called the triplet codon. Now, from the perspective of language, the A's and C's and T's and G's of DNA and the corresponding RNA nucleotides are the letters of the alphabet. And the codons, made up of groups of 3 bases each, are the words. There are about 70 different codons occurring in nature, and they code for only about 20 different amino acids that go into all the proteins that make up living things. The functional unit of DNA that codes for one protein is called a gene, and one may think of a protein in this metaphor of language then as a sentence. So we go from alphabet to word to sentence in the realization of the structure of proteins. It turns out, though, this is not just a metaphor. The genetic code is not simply like a language. It is a language that meets all of the formal requirements for a medium of storing and communicating ideas. And in fact, many of the characteristics of human languages can be found as well in this molecular language. One is a very strict dependence upon the proper grouping of characters of the alphabet to form words. For example, when a change in the DNA sequence occurs, this is called a mutation. And the simplest form of mutation is called a point substitution. That is, a single nucleotide of one sort is substituted for another. For example, an adenine for a thymine. So if we take this sentence as indicative of a molecular language, a point mutation here might, for example, replace one letter of one word, but you see a very dramatic change in the meaning of the sentence. I chose this sentence, of course, because it has special meaning for those in the Geisel Library and us generally on the UCSD campus. Another form of mutation, well, actually before I would mention that although this looks like a simple and somewhat amusing change, there is a very compelling set of circumstances in the real world where this kind of change has devastating consequences. For example, it is the change of just one nucleotide of the DNA sequence that changes the hemoglobin molecule in human red blood cells so that that those cells begin to sickle. That is, they twist and shrink, causing sickle cell anemia, which is a devastating disease that afflicts millions of people worldwide, causing pain and suffering and death. So these very subtle changes in the DNA sequence can have very dramatic changes in the organisms that sustain them. Another form of mutation is an insertion. That is the general idea of inserting just a single nucleotide, or perhaps more than one, into this DNA sequence, so this sentence, if you will. So for example, here, if we took this familiar sentence and inserted just a single letter into one of the words, two changes occur. One is that individual word takes on a much different look, and in a protein synthesis motif may produce a different amino acid there. But it pushes the rest of the characters down. Remember, we're talking about three-letter words, and we shift the reading frame, so-called frame shift mutations, that can reduce the rest of the sentence to utter nonsense, except that, ironically, look, we've created a new word here just by chance that didn't occur before, and that happens in nature as well. These insertion, and as you might imagine as well, deletion mutations that shift the frame at which the DNA is read into proteins, have devastating consequences because by and large they produce defective proteins. Or if the word stop happens to randomly appear in the sequence, then they may produce no protein at all. So you may be asking, well, if we have the genetic code, why can't we just read the sentences and figure out what the book of instructions of living systems is saying? Well, there are two answers to that question, and the first one is that we are just now getting the whole book to look at for a variety of organisms, including human beings. These reports from the mid-1980s were the beginning of a major multinational project called the Human Genome Project, whose goal was to get the complete 3 billion alphabetical characters that make up the sequence of the DNA in each of our cells. Just this year, it was announced that there was a, quote, working draft of the human genome had been obtained by work not only of thousands of government-sponsored researchers, including those here on the campus at UCSD, but also private companies such as Celera Genomics, interestingly headed by a UCSD alumnus, Craig Venter. The confirmation of the sequence, and you see here actually, if you can read that, it is accomplished. The human genome has been assembled. Well, there's a little hubris here because we're not quite done. But we are at a point where we're within the copy editing and proofreading stages of having at least one copy of the genome. And in the next several years, we will have all of the pages of the Molecular Book of Humanity available to read. But now that the string of characters is available, there are other more important challenges. How many of you recognize this famous piece of literature? Anybody? Well, I've made it a bit harder for you than it might have been, but if I highlight it the sequence of interest there, some of you will recognize it. And in fact, if I add back in the white space, it becomes much more understandable and readable. It gets a lot clearer if I'm able to translate the sequence into the words. And finally, if all else fails, I'll just add a few more words to set the context. So the other problem with reading the book of living cells is that it has no white space between the words. And the important messages are buried among long strings of DNA for which we do not know the function. These intervening sequences, as they are called, have been labeled by some as junk DNA because there is, as yet, no evidence that it has a function. Just last month, one of the headlines was that this working copy of the draft sequence of the genome may be up to 97% non-coding or junk DNA. Now, this doesn't mean it's trash to be discarded. If it is junk, it may be junk in the sense that you probably have some junk in your closet or in your attic that is valuable enough that you keep it around because you might use it someday, even though you're not using it now. And so researchers have postulated that perhaps these non-coding or intervening sequences may contain the biological equivalent of spare parts that can be accessed over time in evolution. Others think that if the expressed parts of the genome are like computer hardware, well, maybe those intervening sequences, that's software that tells biological systems how to behave. We simply don't know the answer to this question right now. And it turns out the problem of reading the language is even more challenging than that, because in higher organisms, we're one of them, genes are not composed of single uninterrupted groups of nucleotides. The words are expressed into proteins, but the DNA sequence is broken up with sequences called introns, that is, non-expressed, non-coding pieces that are inserted amongst the pieces that are finally expressed. It is as if every sentence has nonsense words inserted into it. And so you have to either get rid of those words or jump over them in order to read the message. Now, all of these hidden patterns and complications in the molecular language means that molecular biology now has a new essential tool. Gregor Mendel planted peas, and biologists used test tubes and petri dishes and funny-smelling chemicals. But it turns out you can't do this kind of science today without computers. Even if all we wanted to do was browse the human genome, just read it from front to back, it would comprise, if printed in 10-point type, about 200 phone books the size of San Diego's Yellow Pages. As a result, the only reasonable place to keep this kind of data is in databases. And this is a record from the International Sequence database called GenBank. It's the largest publicly available database of these A, C's, T's, and G's that represent the sequence of DNA genes and the literature from which they have been extracted. And for more than 20 years, researchers have been putting data into these databases, which now contain billions of alphabetical characters from millions of pieces of DNA and RNA from literally thousands of different species. It turns out this is a very important GenBank record that has a special place in the history of biology and also has a connection to UCSD. Because this is the GenBank sequence of a gene called the human C-cis proto-oncogene. Now, oncogenes are a special form of gene that, when switched on, cause cancer. That is, the uncontrolled proliferation and growth of cells. They were first discovered in animal systems back in the 1970s. Imagine our chagrin when in the early 80s we discovered that normal human DNA in patients who had no cancer contained cancer-causing genes. And the question is, why would we be carrying around with us the seeds of our own destruction? The answer to this question came from two groups, one led by Russ Doolittle here at UCSD and another group in London, who found by computer matching of the sequence of the C-cis proto-oncogene that it matched exactly a normal growth and development gene called platelet-derived growth factor. In fact, you see that here in the description of this gene. And suddenly it became clear that cancer might be a normal gene just switched on at the wrong time. This kind of fundamental insight was an early example of a field of science that has now come to be known as bioinformatics. That is the application of computer technologies and information science principles to analyze living organisms at a molecular level. UCSD has just launched a graduate program in bioinformatics, and starting next year will be one of the first universities in the country to have an undergraduate major in bioinformatics. And this will be just a wonderful education in both the principles of biology and the principles of computer science, and how to use computers to read this molecular language of living organisms. But just having the sequence of the genes is only the beginning. This is a cartoon actually that's more than 5 years old. It was sort of looking into the future. The caption is, "The good news is we have the human genome, and the bad news is the computer alphabetized it," right? Well, so now we really do have the human genome, and we have to change the caption of this because we've been careful not to alphabetize the sequence. And the new caption is, "The good news is that we have the human genome, and the bad news is it's just a parts list." That is, knowing the structure and knowing the sequence of genes really tells us nothing about how they are switched on and off and how they are orchestrated in cells and tissues with literally tens of thousands of genes working in coordinate patterns to carry forward the metabolic machinery of the individual cells and also do the specialized functions that each of our tissues and the tissues of other organisms have. For these kinds of questions, we get help from a new kind of science that also is making great strides here on campus, and that's called functional genomics. Using robotic technology to attach really thousands of tiny little pieces of specific DNA representing a marker for an individual gene, and we can immobilize those on either glass slides using these robots that print the DNA with steel pens, or on silicon chips. We can now measure whether or not at any given point in time tens of thousands of genes are switched on or not, and how active they are. The output of this kind of experiment looks like this in its raw form, where each one of these dots represents whether an individual gene has been switched on, and the brighter the dot is, the more copies of messenger RNA it's making, because this is really a messenger RNA detector, if you will, and the black areas represent genes that are switched off. This kind of data is an area, interpreting this kind of data is an area that is very rapidly progressing. And as you can tell, there's just too much of it. There's no way by inspection to tell what all of these dots mean. So once again, we rely upon a computer. In fact, this particular problem has been likened to trying to drink from a fire hose because of the volume of the data of thousands of genes taken in a snapshot at once. It points to an era, however, where we could use this kind of expression profiling, very importantly, to transform healthcare, where instead of having diagnosis by opinion, in my opinion, which is one of the first phrases we teach in medical school, we could substitute actual molecular data of knowing exactly what's going on in your cells. And by the same token, knowing what the diagnosis is, instead of trial and error treatment, which is we're going to give you this medicine, which works for most people, and we don't really know if it'll work for you, but give it a try. And if you have a problem, call me. Instead of that kind of trial and error reasoning, we could actually plan with certainty to know at a molecular level this is exactly the medication that you need to affect your particular genes, not generally human genes, but just your specific genes. So all of this points to the fact that we are poised at a truly exhilarating time in the science of biology, and all of you who are here working at UCSD to support our missions of research and education are doing more than you know to broaden the base of our understanding and to reduce the burden of suffering and sickness worldwide. And for those of you at the Preuss School who have an interest in science, I hope you'll look into biology and into bioinformatics, because we've only just started this era of genome-enabled science, and it will go on for decades, building and discovering fantastic new things about how normal cells work and how disease affects them. Plus, it doesn't hurt that people who know both biology and computers really well get very nice jobs and get paid a lot of money. In the future, any one of you might be the person who finds the molecular words that prevent cancer, or perhaps genetically engineers a new kind of banana that is not only nutritious but also cures malaria in third world countries, or perhaps finds a way to save yourself and others from heart disease. So in the spirit of this lecture series, I hope that you will agree that viewing biology as literature is a science that really matters. And I thank you.